experiment was on the effect of color on photosynthesis. Our scientific question was, will spinach leaves produce a different amount of oxygen through photosynthesis if different color lights are used as a light source? Our hypothesis was, spinach leaves will produce a different amount of oxygen through photosynthesis if different color lights are used as a light source. The dependent variable was the number of discs floating per 20 minutes, and the independent variable was the color of the light filter. We believe that the independent variable will affect our dependent variable because the spectrum of visible light that the sun gives off includes different colors. And because plants require sunlight for photosynthesis, they should be able to absorb some of these colors more effectively than others. Some colors may be reflected off the leaves like green, and others may be absorbed more effectively. Our replication was repeating the experiment twice over 2 days. Our control was a light bulb with no color. This was our positive control because we knew it was effective. Our negative control was green light because we knew it cannot be absorbed by plants. In this experiment, we will put a different color paper under the light and after a set period of time, count how many discs are floating, which is a measure of the oxygen produced by photosynthesis. We predict if our results support our hypothesis, then the number of floating discs will be the most after the plant has been exposed to red light. If our hypothesis is false, then the number of floating discs will not be the highest after the plant is exposed to red light. Our results showed that the most discs floated when the spinach discs were under red light. The least amount of discs floated when they were exposed to green light. The second most discs were floating when there was no color and then the orange light. When exposed to blue light, the second least amount of discs were floating. Looking back at our hypothesis or question we posed at the beginning of the experiment, our hypothesis stated that different light filters would cause spinach leaves to produce different amounts of oxygen in photosynthesis. Our hypothesis was supported because the rates of photosynthesis were dramatically different based on what light filter was present. For example, the percentages of floating discs after 20 minutes under red filter light was 70%, 60%, 80%, and 60%, while the percentages of floating discs after 20 minutes under the green light were 20%, 10%, 6.6%, and 20%. Here's a picture of our graph, which shows red, blue, orange, green, and no color filter. As you can see, this blue line says the average, and the average for the red color light was more than the green color light and the rest of them. Our results did correspond to the prediction we made. The different light filters did affect the rate of photosynthesis of the spinach discs. The discs with a green filter light had a very low rate of photosynthesis, while the discs with the red filtered light had a very high rate of photosynthesis. Our prediction was based on the idea that there is an optimum wavelength of light that can be best absorbed by plant pigments. We expected all the tests other than the green light filter to have some rate of photosynthesis. This is because when we looked at plants, we see green. Therefore, green is the only wavelength not absorbed by pigments in the plant and is therefore reflected. Our results make sense because the green had the lowest rates of photosynthesis and the other colors had a higher rate of photosynthesis. The number of discs for each color then gave us an idea of the optimum color filter for the rate of photosynthesis. This is a data table showing you our results. It is the percent of discs floating in each of our 4 trials and the average at the end. That's a picture of our setup of the experiment. From our textbook, we know that photosynthesis depends on the absorbance of light by the pigments in the plant's leaves. The colors that we used are different wavelengths of light being absorbed by the plant. The pigments, like the ones we observed in our chromatography lab, are the same ones we worked with in this lab, such as chlorophyll A, chlorophyll B, and beta-carotene. Chlorophyll, according to the biology site at Westfield State University, are the most common and highly important pigments found in plant leaves. The only color they do not absorb is green. In contrast, they're reflected, and that is why most plants are green. Based upon this, the absorption rate of chlorophyll A can be almost directly related with the reaction rate of photosynthesis. Our results are supported by all this information because, as we can see, the Petri dishes with the green wavelength absorbed no more than 20% and absorbed most in red and orange. Although our results did support our hypothesis, there were some inconsistencies in our data. The information we gathered was mostly qualitative, with amount of disk floating or percent of disk floating instead of a direct amount of oxygen produced illustrating the rate of photosynthesis. Some of our blenders were the amount of light distributed to each Petri dish, the size of our heat sink, declaring whether a disk is floating or still at the bottom, the height of the lamp, the exposure of Petri dishes with bicarbonate and disk to light before the testing began. Some of our errors were the difference in exposure to room lighting, discrepancy in spinach disc in thickness, size, shape, and amount of color. If we had the opportunity to repeat and extend this experiment, we would do much more replication. We found that the numbers in each trial did not correspond with each other in every occasion. Replication would make the data more reliable or credible. It is clear to see from our data that replication is necessary to eliminate experimental error. We would also try to create an experiment in which we could get true qualitative results instead of the partially quantitative and qualitative results. From our results, we can conclude that the red light filter was the optimal wavelength of light for the absorption of light by pigments and leaves of plants. We also have supported the hypothesis that the green light filter is the least absorbed wavelength of light for spinach leaves. If we designed an experiment based on our results, it would be what color glass on a greenhouse optimizes the growing conditions for plants inside the greenhouse. We would design this so that we could see which color filter of light worked best for growing plants in a greenhouse. This would be a larger scale version of our experiment, seeing how the results can affect real life situations. Based on our conclusion, we can see that there is an optimum color filter for the reaction in photosynthesis. This investigation could change the way greenhouse farmers approach the growth of their plants. The object of growing plants in a greenhouse is to optimize the growing conditions to maximize growth. We could apply our conclusions about light filtering to greenhouse conditions by measuring the growth in plants in greenhouses by filtering the light inside of them. Because we concluded that red light was beneficial for photosynthesis, we hypothesized that red light will be best for plant growth. By increasing the rate of photosynthesis, the plants could be lowering the amount of CO2 in the atmosphere and producing more oxygen, countering the greenhouse effect of global warming. All in all, this is our results and discussion of our experiment and all our trials.